Pharmaceutical facilities rarely dedicate an entire building to a single drug. Instead, they operate in campaigns. A factory might manufacture a cardiovascular medication on a Monday and an entirely different formulation on a Thursday, utilizing the exact same hardware. The physical scale of this hardware is massive. We are looking at interconnected stainless steel cylindrical tanks and piping systems designed to hold thousands of liters of product at a single time. The risk here lies at the molecular level. Traces of the active pharmaceutical ingredient, or API, from Monday's batch can easily cling to the walls of these vessels. To the naked eye, the stainless steel might reflect light perfectly, but a vat that looks spotless can still harbor enough active residue to trigger a severe allergic reaction or toxicity in the next patient. To prevent cross-contamination, manufacturers must scientifically validate the efficacy of the cleaning process. The protocol begins the moment a production run ends. Facilities enforce strict holding times. The maximum allowed period equipment can sit dirty before drying residues become permanently baked onto the metal. Operators then physically disassemble the hardware, exposing transfer pumps, hidden seals, and valves. Scrubbing by hand introduces human error. To remove that variable, the industry relies on automated clean-in-place, or CIP, systems, handing the responsibility of scrubbing over to automated chemistry. The CIP system unleashes a storm of pressurized fluids and dissolving agents, blasting the metallic surfaces to strip away invisible layers of residue. The system logs every variable in real time: the exact volume of the water, the heat of the cycle, the force of the pressure, and the precise concentration of the detergents used. But the detergents themselves introduce a new variable. Strong acids and alkalis can leave behind their own chemical breakdown products, turning the cleaning agent into a contaminant if it isn't thoroughly flushed from the system. Mechanical cleaning provides a pristine surface, but the protocol requires proof that every hidden residue has been neutralized. To prove the tanks are clean, analytical chemists assume the process failed. They design sampling plans that actively hunt for contaminants in the worst-case locations. Through direct surface sampling, analysts target the most difficult-to-clean geometric features inside the machinery. They go after the weld seams, the mixing blades, and deep internal crevices. They use specialized swabs made of inert materials that won't interfere with testing. The analyst rubs the swab in a strict cross-hatch pattern over a specific surface area, applying precise pressure to trap any remaining trace molecules. The primary limitation of a swab is physical reach. An analyst cannot push a probe deep into a narrow, 100-foot network of internal plumbing. To cover the inaccessible piping, they use indirect sampling, or the rinse method. By capturing a sample of the final flush of pure solvent running through the entire system, they can detect ambient, system-wide traces of residue. This visual compares a normal therapeutic dose to the maximum allowable carryover limit, often just 10 parts per million. Watch how this scales down to a razor-thin line. Industry rules mandate no more than 0.1% can appear in the next product. Detecting a fraction of a percent requires highly specialized analytical methods. Instruments like high-performance liquid chromatography, or HPLC, are calibrated to achieve the sensitivity needed to isolate and identify stray molecules. When the HPLC prints its results, the verdict is final. In cleaning validation, cleanliness is a mathematically proven threshold derived from hard data. The validation process culminates when that laboratory data meets final human oversight. The quality assurance manager performs a final, visually clean inspection. No residue can be visible on the equipment, providing a common-sense verification to complement the microscopic data. Every step of this routine, from the swab pattern to the HPLC calibration, is dictated by good manufacturing practices. These are rigorous regulatory frameworks established and overseen by global bodies like the World Health Organization. The final validation report compiles the test results alongside secure, computer-generated audit trails and authorized electronic signatures, ensuring that every data point is traceable and unaltered. Cleaning validation is an obsessive scientific discipline. It operates entirely behind the scenes to guarantee that every pill, vial, and syringe contains exactly its intended medicine and absolutely nothing else.